If a neuron is going to communicate information from one body part to another, those action potentials have to travel down the axon from where they were triggered at the axon hillock to the axon terminals so that neurotransmitters can be released on the next cell and cause an action to occur. Unlike we saw with the graded potentials, action potentials don't die out, so they keep their strength as they spread across the entire membrane of the neuron. In a neuron, an action potential can only propagate in one direction. So propagate is this moving down the axon of that action potential. So the depolarizing from one area to the next, to the next, to the next. So think of that as you would think of with dominoes. Once you push one domino down, the others are going to fall. That also brings us to the point of stimulus strength. So think back to that domino analogy again. If you have dominoes lined up and you push the first domino in the line of dominoes hard enough, it's going to fall, and then it will knock down the second domino, which will knock down the third, and so on until it reaches the end. This is known as the all or none action of propagation of an action potential. So a strong enough stimulus is going to cause an action potential to fire. Just like if you push the domino strong enough, it's going to cause the dominoes to fall all the way to the end. So regardless how hard you push that domino, the effect is not going to increase in intensity. So the same thing with action potentials. A stronger stimulus does not increase the firing of the action potential in strength, but it can cause more action potentials to be fired. So there are some factors that affect the speed of propagation. Remember, propagation is simply the movement of that wave of depolarization down the axon. So axon diameter can affect the rate in that larger diameter axons propagate action potentials faster than smaller ones due to their increased surface area, so more sodium gates open and more depolarization occurs. Axons propagate action potentials at lower speed when they are cool. That's one of the reasons that you put ice on a painful area, because that localized cooling of a nerve can produce sort of an anesthetic effect because it slows the rate of those action potentials propagating down that axon, and the amount of myelination. So we've talked a little bit about myelination. Axon potentials propagate more rapidly along the axons that are myelinated than along unmyelinated axons because of something we call saltatory conduction, which we'll talk about in just a moment. There are two types of propagation: continuous conduction and saltatory conduction. The type of action potential propagation described so far has been continuous conduction, which involves a step-by-step depolarization and repolarization all along the membrane of that axon. In continuous conduction, ions flow through the voltage-gated channels in each adjacent segment of that membrane. So the action potential propagates a very short distance in a few milliseconds. Continuous conduction occurs in unmyelinated axons and in muscle fibers. So here you can see in version A, the action potential is propagating along the axon. So in 1 second, we are depolarizing this area of the membrane, and that's going to cause adjacent voltage-gated ion channels to open here in the membrane, so more sodium comes in, and now this part of the membrane depolarizes, and that continues. So after 5 milliseconds, you can see the action potential has only traveled a short way down the axon, and in 10 milliseconds, still a very short distance. In comparison, if you look at the second part of the image here, we see what is known as saltatory conduction. So action potentials propagate faster along myelinated axons. So if you compare part A and part B, you'll see the action potential propagates a lot farther along the myelinated axon in the same period of time. Okay, So we call this saltatory conduction. Saltare means leaping. So it was once thought that the way that this action potential is moving looks to be moving from node of Ranvier to node of Ranvier. So saltatory conduction is the special way of propagation of the action potential that occurs along myelinated axons because of the uneven distribution of the voltage-gated channels. So there are few voltage-gated channels present in regions where there's a myelin sheath covering the axon. By contrast, at the nodes of Ranvier, there's not a myelin sheath, so the axolemma has a lot of voltage-gated ions. So the current is carried by the sodium and potassium that flows when the membranes open, mainly at the nodes. So if we look at part B of this image, so we have a threshold stimulus causing depolarization. So we depolarize the membrane here, and then we've got a node, we've got a myelin sheath, right? So here's the Schwann cell, node of Ranvier, node of Ranvier, and node of Ranvier. Remember, the nodes of Ranvier are the areas that are not covered by the myelin sheath. So from this initial 1 millisecond after depolarization starts, and then to 5 milliseconds, look how much further the wave of depolarization has propagated. And that's because once an area of the membrane depolarizes, that flow of electrical current basically just sort of shoots through that part of the axon that is covered by the myelin sheath. Because there are fewer ion gates there, you don't have to have all of that time for the sodium to rush in and depolarize and the potassium ions to start pumping out. So only the areas in the nodes of Ranvier are depolarizing, and therefore the action potential propagates or travels down that axon much faster in myelinated axons.